it's me Chiku from Chiku Study Corner, and today we'll be dealing with the next part of Biology Class 12 CBSE Chapter 10, that is Human Health and Welfare. This is part 2 of this chapter. Please make sure you watch part 1 because part 1 is very important and it's a continuation. So let's get into the video. In the previous session we discussed about what the industrial, the household, etc. what the use of microbes or the role of microbes in these sectors. Now let's see what is the role of microbes in what? In human body. So, to talk about the human body, in our human body, there are some certain places where these microbes are colonized. So, let's see. Human body colonizes different microbes in different parts of the body, like in our guts, in our skin, in our buccal cavity, in our esophagus, in our intestine, etc. So, every portion has a different type of microbe there. Because if there's these would help us in our bodily functions. Okay. So, next one is the most important, very important microbe in our body is gut microflora. Okay. So, this gut microflora has two properties or two determining what you say significant properties. That is, first one is they secrete a substance that is very good for digestion. So, what is happening? What does this do? This helps in digestion because they secrete a particular substance. Okay. The next one is it prevents other microbes from colonizing in our body. So, you just think there can be some other microbes in our body which are harmful, or anything displaced or anything not in its place is not good for us. If you say microbe that has to be in our buccal cavities and our stomach, what would happen? There would be an adverse effect. So, what is that? It prevents the other microbes from colonizing in different parts of our body. So, the next one is stomach microflora. So, what is stomach microflora? They are the microbes that are found inside the stomach. The example is Streptococcus and Staphylococcus. So, these two microbes, they can withstand the acidic conditions of the stomach. Or else how would they live inside this? I told you they are omnipresent. They can live in any conditions, in like high temperatures, in low temperatures, in acidic, in alkali, in any medium, even inside metals you find what? Microbes. So that is to talk about what? Stomach microflora. The next one is intestinal microflora. So that is enterobacteria. Okay. So that is the microbe that is there inside our intestine. What happens? It helps in digestion and absorption. How does it help? It increases the efficiency of digestion. So it produces something that will help or that will fasten the digestion processes. So now we discussed about two of them which helps in digestion. What was that? It was gut microflora and intestinal microflora, that is enterobacteria. Okay. The next one is, viruses. Viruses are used as a vector for transmission of required gene during recombinant of DNA. Okay, in DNA technology. Okay, so that is what viruses are being used. See, even viruses are microbes. They are used for what? The transmission of the gene for DNA recombination in that technology. Okay, so now we discuss about microbes in the treatment of the sewage water. So, microbes in sewage treatment. So, what is sewage? The waste water that is given out from our houses, from households, that's known as sewage. Okay. So, let's see. There are various steps for what? This sewage treatment. So, two steps are primary treatment and secondary treatment. So, first we will discuss about primary treatment. Primary treatment is the physical removal of solid particles from the sewage water. That is what the primary treatment. There are two steps that comes under primary treatment. The first one is filtration. The second one is sedimentation. So, what is filtration? that is these large molecules or large particles like stones, pebbles, plastics, papers, etc. have been filtered, and then the effluent that is given after the filtration is allowed to settle. What is sedimentation? The settling down of what particles, that is we learned about solute and solvents, the same way settling down of solute particles under the influence of gravity. Okay, that is, so actually a colloid only would show this property. Okay, and after sedimentation the effluent is transferred to the secondary treatment tank. Okay, this is a primary treatment tank. Now it's moved to the secondary treatment tank. So let's see what is the secondary treatment. So the effluent from the first tank is moved to the primary tank, and therefore what happens there? Aeration takes place. What is aeration? Continuous supply of oxygen. Why do you give continuous supply of oxygen? When you give continuous supply of oxygen, what happens? The microbes grow in it. What kind of microbes? Anaerobic aerobic microbes grow there. So after the aeration, these microbes, what would they do? They would feed on these organic matters and hence form flocks. And what happened? Due to the formation of flocks, the BOD decreases. What is BOD? Biological oxygen demand. Due to the decrease in biological oxygen demand, there are some things that arises. So what after this happens, after BOD is reduced, what happens? There is a settling tank. After this is all done, now the microbes have eaten all the biological waste inside that, and then what happens? This is moved to the settling tank, from the secondary tank to the settling tank. And what happens? The flocks inside that, the biological flocks inside that the flocks, what the microbial flocks, okay, because now there is a decrease in oxygen, and therefore these would undergo sedimentation. And after undergoing sedimentation, that sediment is called as activated sludge. Okay, this activated sludge is now divided into two. They take a small portion and they add it to our aeration tank. Here we had an aeration tank, no? They add it to the aeration tank to act as an inoculum. Okay, sorry, inoculum. So we learned what is inoculum in lactic acid. Yeah, inoculum is a starter or something that fastens the process. So the first part as a inoculum, and the second is the major part, the huge part is moved into the anaerobic sludge digesters. What happens there? During this process, it releases a huge amount of gases. That is, one is CH4, that is methane, carbon dioxide, and hydrogen sulfide. So these are the gases that is being released. So, in our country, the Ganga Action Plan, what does it say? It says that you have to have more sewage treatment plants. Why is that? Because only treated water should move to the river, because Ganga is being polluted so badly. Before it was believed that if you have a dip in Ganga, your diseases, all your sins will be washed away. Now if you go take a dip in Ganga, diseases would come because it's so dirty. That is why our government put forth Ganga Action Plan for to save our river Ganga. So what was that? The government wanted to increase the number of sewage treatment industries. Therefore, what happens? These treated water would only be given out to the rivers, and there is no harm for the life inside the water, and there is no harm for life outside the water. There are many people who take water from that, who take a dip in it. So there is so many that can cause a very, very bad adverse effect. So now let's talk about the treatment, the sewage treatment. Now we'll discuss about microbes in gobar gas. Gobar gas is also called as biogas. Okay? Biogas is an alternative that is used in many homes for cooking and other sources of energy. Okay, let's see. Biogas is obtained by let's take, there's a bacteria which lives anaerobically on cellulosic materials. What is cellulosic materials? Dead and decaying matters of plants and animals. Biodegradable, things which are there is known as cellulosic materials. So that is how biogas is formed. These bacteria, what would they do? They would extensively produce CH4, CO2 and H2, that is carbon dioxide, methane and hydrogen gases. So actually CH4 is the highest amount, okay? And okay, let's see. These CH4 producing bacteria are collectively called as methanogens. Okay, an example of methanogen is methanobacterium. Okay, so that is the example for what a methanogen. So that is a talk about what is microbes in Biogas. Now let's see the last one, microbes as biofertilizers. Now nowadays the farmers are forced to carry out their farming in the basis of organic farming, because now the chemicals have destroyed the soil, the plants, etc. The increase in the use of chemicals have destroyed almost everything. So there comes the biofertilizers. Biofertilizers are obtained from these biodegradable substances that are found around us. You can make your own compost, which can be used as fertilizers for your own plant. So and over here what happens is, there are various microbes who act in the role of a biofertilizer. First one we have to learn is rhizobium. We have learned in our lower classes. They are there in the roots of legumes. What do? They fix nitrogen. Okay. Other than rhizobium, there are some other organisms which fixes nitrogen, that is called as azapyrium and acetobacter. Okay, these two are also examples of what? Nitrogen fixing bacteria. Okay, these I told you it can be, biofertilizers can be microbes, and these microbes can be bacteria, fungi or even cyanobacteria. Okay, so let's see, fungi plus the root of huge trees, what would they form? They will form a symbiotic relationship and they form something known as mycorrhiza. Okay, and what does fungi do? It absorbs phosphorus from the soil. So does fungi play a role? Yes. Rhizobium is a bacteria. Does that play a role? Yes. So let's see. Now our cyanobacteria. What does it do? It fixes almost everything that is required for the plant. It fixes many things. So that is also talk about what is microbes as biofertilizers. So friends, we have come to the end of this chapter. I hope you might have understood it. If you understood it and liked it, please give my video a huge thumbs up and subscribe to my channel if you're new to it. And share my video with your other friends. So see you on my next video. Till then, bye-bye.